Hello, this is Dr. Li Hua Stephan. Today I'm going to talk about ligation and transformation. The learning outcome for this lecture include define and explain how ligation and transformation work. Describe methods of introducing foreign DNA into prokaryote system. Calculate ligation ratios based on length of vector and insert. List appropriate controls for a typical transformation of a ligation mixture. Traditional gene cloning usually involves the following steps. First, we need to isolate our gene of interest, which usually serves as the insert. Then, we will select the vector, which usually contains a selectable marker and a multi-restriction enzyme site. We then digest both insert and vector with restriction enzymes. Following the restriction enzyme digestion, we will perform a ligation reaction to join the two pieces together to form the recombinant plasmid. Following ligation, we can transform the recombinant plasmid into competent E. coli cells. We can then plate the transformed E. coli cells onto a selection plate and screen for the positive clones. Today, we are going to focus on ligation and transformation. Ligation is a joining of two DNA fragments through the action of DNA ligase. The DNA ligase catalyzes the formation of a covalent phosphodiester linkage between 3' hydroxide and 5' phosphate on DNA. A cofactor like ATP is required for this process. We need to notice that DNA strands do not spontaneously ligate. Even the strands have complementary cohesive end. DNA strand must be present. DNA ligase must be present for ligation to occur. There are several rules for ligation. In general, we will set up the reaction in small volume of 10 to 20 microliter so that the cohesive ends will not be too dilute. We usually use 50 nanogram of DNA for average size vector. The ideal amount of insert to use can only be determined in individual base, but in general, we will try to add in the insert at 1:1, 1:3, and 1:10 vector to insert molar ratio. For example, if you are using 3 kb vector and a 1 kb insert, if you want to use 1:1 vector to insert molar ratio, you want to use 15 nanogram of vector with 16.7 nanogram of insert. If you want to use 1:3 vector to insert molar ratio, you want to use 15 nanogram of vector with 15 nanogram of insert. If you want to use 1:10 vector to insert molar ratio, you want to use 15 nanogram of vector with 167 nanogram of insert. We usually use the following formula to calculate how much insert we need to add to a ligation reaction. The insert to vector mass ratio multiplies the desired molar ratio of insert and vector will equal the ratio for the amount of insert and vector that should be added into the reaction. For example, you have 15 nanogram of 4 kb vector and you want to ligate your insert, which is 2 kb, at a 10:1 insert to vector ratio. How much insert you need for this reaction? Let's use the equation above to solve this question. First, let's put the insert and vector mass into the equation, which is 2 kb and 4 kb. Next, let's put the insert and vector molar ratio into the equation, which is 10 and 1. Finally, on the right side of the equation, let's put the amount of vector that we want to use, which is 15 nanogram. We can use X to present the amount of insert we need for this reaction. After calculation, we can get X equal 250. So, we need to use 250 nanogram of insert for this ligation reaction. The stock concentration of insert is 25 nanogram per microliter. What volume shall we use? We can calculate the volume desired for this reaction using the desired volume of insert, that is 215 nanogram, divided by the concentration, that is 25 nanogram per microliter. We can then determine we need to use 10 microliter of insert. Now, we know how much vector and insert to add to a ligation reaction. The next thing we need to add is a ligase buffer. The ligase buffer contains the necessary salt and ATP to allow the ligase work appropriately. Because ATP is unstable, we need to keep the ligase buffer on ice. Finally, we need to add ligase. For most reactions, 1 microliter ligase is sufficient. However, sometimes for blunt end ligation, a higher amount of ligase is desirable. Now, all components have been added to this ligation reaction. The next thing we need to do is to set up the ligation reaction at the appropriate temperature. The temperature of ligation is usually determined by the optimal temperature of a ligase activity and the melting temperature of DNA ends. For short stretches of DNA, we can usually calculate the melting temperature to be 4 degrees times the number of G and C plus 2 degrees times the number of A and T. Above this temperature, the DNA will not anneal. Usually, for compatible cohesive end, the ligation reaction is incubated at room temperature for 2 hours or overnight at 16 degrees Celsius. For blunt end reaction, the ligation is incubated overnight at room temperature. Now, we have learned what a ligation is and how to set up a ligation reaction. Following the ligation reaction, the insert and vector DNA will join together to form a recombinant DNA molecule. Next, we want to introduce this recombinant DNA into host cells for selection and further propagation. Usually, we use a competent E. coli cells as a host. The ability for a bacterial cell to take extracellular naked DNA from its environment is called a competency. The natural competency of E. coli is very low. The E. coli cells need to be made competent for transformation. To make the E. coli cells competent, we usually treat the E. coli cells in log phase of growth with a divalent cation, for example, calcium chloride. Pre-made competent E. coli cells are also commercially available. For more information about the mechanism for preparing E. coli cells competence, you can view the animation at the following website. To set up a transformation reaction, first, we will mix the DNA with the competent E. coli cells. Usually, we will add 1 to 5 microliter of the ligation reaction mix to the competent E. coli cells. This process needs to be performed on ice. Following incubation on ice, we will briefly heat shock the cells to allow the DNA to enter the E. coli cells. Following heat shock, we will return the cells to the ice. This process will make sure the DNA enters the E. coli cells. Following transformation, the cells will then recover in rich broth for 30 minutes to 1 hour. Finally, we will plate these transformed cells on an antibiotic selection plate. The ligation mixture may contain different kinds of molecules. However, only the circular DNA is transformable into E. coli cells. The linear DNA can enter the cells, however, it cannot be replicated. Supercoiled DNA transforms better than non-supercoiled DNA due to its compact shape. Therefore, undigested supercoiled DNA vector will transform at a higher efficiency than digested vector with a ligated insert. Once the transformed cells are plated on a selective media, each colony represents a clone. On the right, we're showing how a plate will look like from a transformation. The red arrow is showing one colony on this plate. This means that every cell in this colony has the same plasmid in it. In our experiment, we will ligate the PCR amplified and digested Campari DNA with the pN34 vector. We will then transform this ligation mix into E. coli cells. Because the pN34 vector contains the cannomycin-resistant selectable marker, the successfully transformed E. coli cells will therefore be able to grow on plates containing the antibiotic cannomycin. This is a result we will expect. However, what if the next day we find an unexpected result like this, with no colonies? If this happens, clearly something is wrong. Things can go wrong during ligation and transformation include contamination of DNA, bad ligase or ligase buffer. The competent E. coli cells may be bad, or the antibiotic may be inactive, or the digestion of the vector before the ligation may have been incomplete. In order to perform a careful experiment, we need to set up several controlled reactions in addition to our experimental reaction. In our case, the experimental reaction is a digested Campari PCR fragment with a pN34 vector. Following the ligation, we will expect a moderate efficiency of transformation and growth on the cannomycin plate. In the case of unexpected result, we can investigate the controls. This control can tell us whether the unexpected result may either due to bad competent cells or bad ligase. To control for competent cells, we will transform the uncut PET41A plasmid, which also contains a cannomycin-resistant selectable marker. We will expect this uncut supercoiled DNA to have a higher transformation efficiency and a higher number of clones on the plate. If that did not occur, we can either suspect the E. coli cells are not competent or the cells were dead. In order to control for ligase, we will use a single-cut PET41A plasmid. Following ligation, we will expect a moderate transformation and growth on selection plate. If we observe no clones, we can suspect the DNA ligase went bad. In summary, we hope following this lecture, you are able to define and explain how ligation and transformation work. You can describe methods of introducing foreign DNA into prokaryote system. You can calculate ligation ratios based on length of vector and insert. You can list appropriate controls for a typical transformation of a ligation mixture.